Hello everyone, this is Dr. Nousheen. Today I will teach you first year biology with chapter 4 Bioenergetics. We will start with the topic cellular respiration. When we eat food, the food breaks down into glucose. The glucose goes into the blood and ultimately into the cells of the body. And the cell uses glucose for the formation of energy because cells are doing activities. The muscle cells, when we are moving, the muscle cells are used, they need energy. The heart cells need energy. Every cells in our body needs energy, so every cell make their energy by its own with the process that is taking place inside the cells. Okay, we are discussing about the cellular respiration that is occurring inside the cells. There are two types of respirations. The first is aerobic and anaerobic respiration that is taking place inside the cell. Okay, when we consume glucose okay, before proceeding towards aerobic and anaerobic respiration, I want to tell you that when the glucose goes inside the cell, it breaks down into pyruvate plus 2 ATP molecules are formed. This process is called glycolysis. I want to tell you that this process, glycolysis, I will tell you about this process in detail later, step by step, but I want to highlight this here because this process is similar in both. This is the initial steps. Is similar in both aerobic respiration as well as anaerobic respiration. Okay, now let's continue with anaerobic respiration. What is anaerobic respiration? Anaerobic respiration is also called fermentation. This is also occurring in the cytoplasm, not in the mitochondria. Even the glycolysis, the initial step, the glycolysis also occurring in the cytoplasms. Okay, so after the glycolysis, there is a molecule made which is called pyruvate. Okay, if the process is anaerobic, these are the steps, but if the process is aerobic, there is another steps I will tell you later. Now, if this is a anaerobic respiration, there is a two types of anaerobic respirations according to where it taking place. The one is alcoholic fermentation that is usually occur in yeast and bacteria. Okay, and the one is called lactic acid fermentation that is occurs in muscles. Okay, and some bacteria also doing with lactic acid fermentation. Okay, what happened? The pyruvate, the molecule that we have, two molecules of pyruvate we have after the process called glycolysis. These molecules goes reductions. Okay, reduction. What is reduction? You should know the definition of reduction and oxidation. You should know that whenever the hydrogen ion attached to the molecule, this is called reduction. But when the hydrogen ion removes from the molecule, that's called oxidation. So here pyruvates, hydrogen ion attached to the pyruvate molecule, and it will become lactate or lactic acid. It become acid. The process is called reduction. This lactic acid plus two molecules of ATP which is already made, this is the end result of glucose. Now this product, this is lactic acid, what it cause? It cause fatigue inside the muscles. Okay, जितना ज़्यादा बनेगा fatigue होता जाएगा. Now the second thing is that alcoholic fermentation, that is what is the additional here? There is also occurring like reduction, but before reduction there is called decarboxylation. What is decarboxylation? From the pyruvate molecule carbon dioxide release. Okay, what made acetaldehyde, and then the acetaldehyde, there is reductions occur, and the molecule which is made, which is called ethanol. This is only 2 ATPs are formed. Okay, from the initial step glycolysis. Further, no, this is the end product of the glucose in anaerobic respiration with just 2 molecules of ATP. Now let's move towards the main. This is also the illustrated diagram which I already told you. Now move towards aerobic respiration. Okay, aerobic respiration have 4 stages. Glycolysis, that is similar in aerobic and anaerobic both. there is link reaction which is called pyruvate oxidation. The pyruvate molecule which is formed is oxidized. Okay, what this process I will tell you later. The third step is Krebs cycle, which is also called citric acid cycle, and the fourth is electron transport chain. In this video, I will teach you glycolysis and link reaction. I will make another video for Krebs cycle and electron transport chain later, right? Now, this is a process called glycolysis. Okay, I will tell you step by step. Okay, so once when you have a glucose inside the cells in the cytoplasm, what actually happens, in the cytoplasms, this the glycolysis itself has two phases. The first is investment phase, or which is called preparatory phase. The second is payoff phase. Initially we need 2 ATPs. We will give 2 ATPs for the process to start, but later in the process we will take back this ATPs, okay, because we are doing this process for energy, not to give the energy, okay, but initially we have to give. Okay, so what it actually happen inside the cytoplasm? There is a ATP which is called adenosine triphosphate. Why it's called tri? Because it has 3 phosphates. Okay, whenever what actually happen in the glucose molecule, there are 6 carbon particles. There are 6 carbons. The one phosphate from the ATP removes and it becomes ADP, adenosine diphosphate, and this phosphate group is attached to the 6th carbon of the glucose. So it will become glucose 6-phosphate. Okay, so it will become, that's why it's called glucose 6-phosphate, because the glucose phosphate is attached to carbon number 6, right? This is called glucose 6-phosphate. This is called phosphorylation. Why it's called, this is called phosphorylation? This is the first step in glycolysis. Why phosphorylation? Because phosphate is attached, that's why it's called phosphorylation. The next step is this is the irreversible step. When the glucose 6-phosphate is made, it will not go back to form again the glucose. This is the irreversible step, right? The second step is called isomerization. The second step is called isomerization. What is happening here? There is another molecule which is called fructose 6-phosphate, which is formula wise similar to glucose 6-phosphate. Both are formula wise same, but they are two different molecules according to their structure. Okay, the formula is same but the structure is different. So the glucose 6-phosphate change into fructose 6-phosphate, okay, to another molecule but with same formula, because there is nothing removed, that's just the formula is changed. So this is called fructose 6-phosphate. This process is called isomerization. Isomerization means that there is a changing of molecule from one molecule to another molecule with the same formula, right? Okay, this is the second step. What is the third step? We need another ATP. Okay, we need another phosphate group. Okay, so now we use ATP convert into ATP, and the phosphate is attached to this fructose 6-phosphate. It become fructose 1,6-phosphate. Why it's called fructose 1,6-phosphate? Because the another phosphate is attached to carbon number 1, right? The previously carbon number 6, now the carbon number 1, so it becomes fructose 1,6-phosphate. This process is also called phosphorylation because phosphate group is attached to the fructose 6-phosphate, right? And we also write it here, like we say this is phosphate bisphosphate. Okay, why it's called bisphosphate? Fructose 1,6 bisphosphate, because of 2 phosphates, right? Now next step, what happened? This fructose 1,6 bisphosphate now going to divide into 2 molecules, okay, with 3 carbon each. Fructose have 6 carbons. Now this have like 3 carbon each. So the 2 molecules are made. Their name is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. You know, these two molecules have same formula, but they are different molecules structure wise. Okay, they are structure because of the attachment of hydrogen, carbon. That's why they are called dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. This is called splitting because this fructose 6 fructose 1,6 bisphosphate split. So this is called splitting or cleavage. Okay, now these, although I'm already told you that they have same formula, then what happened, this dihydroxyacetone phosphate convert into the same glyceraldehyde 3 phosphate. So now we have two molecules of glyceraldehyde 3 phosphate. This is the fifth step, and this is called isomerization, right? Now what we have two molecules of glyceraldehyde 3 phosphate here. Before going towards the next step, are you getting my point? Are you understanding well? Just take a deep breath before we continue it. Now the sixth step, what actually happened with this? Now there is some molecule which is called NAD positive, that is present in the cytoplasm. What actually happened, from this glyceraldehyde 3 phosphate, hydrogen ions removes. Okay, and these hydrogen ions attach to this NAD positive and it become NADH. So now, because these are two, because glyceraldehyde 3 phosphates are two, so two NADHs are formed. Okay, and obviously when the hydrogen ion removed, this is called oxidation or dehydrogenation. You know, D means remove, hydrogen means hydrogen, dehydrogenation or oxidation. Okay, so in this process there is if I'm not wrong, so now this will become 1 3. Okay, in this process there is also one phosphate is also attached, so it will become 1,3 bisphosphoglycerate. Okay, it will become, there are two, now there is a two molecules of one, because this is already have three phosphoglycerate. Now and one phosphate is attached to the carbon number 1, so it become 1, because this molecules only have 3 carbons, so it become 1,3 bisphosphoglycerate. Okay, now this is called this process, this step is the sixth step, which is called dehydrogenation and phosphorylation. Okay, now one thing, this is the question, you have to figure it out that from where this phosphate attach. Previously we say that ATP gives phosphate, but from where this phosphate attached to make 1,3 bisphosphoglycerate? This is the question you have to comment me, that what is the answer, right? Now the seventh step is this 1,3 bisphosphoglycerate need to remove the phosphate. Okay, need to remove the phosphate, and I will tell you the answer in the Krebs cycle. Okay, now 1,3 bisphosphoglycerate, this is the actual time to make ATP. Now the phosphate is released and attached to the ATP, and it become ATP, and it will become 3 phosphoglycerate. Okay, one from the carbon number 1, phosphate is removed and attached to the ATP, it become ATP. So now these are actually the two molecules. Now two molecules of ATPs are formed. This is called ATP formation. Now we pay off the investment which we already done previously. Okay, so two molecules of ATPs are formed in this process. Okay, so this is called ATP formation. This 3 phosphoglycerate, the two molecules, is changed into another molecule with same formula, which is 2. What actually happened, just the phosphate group removed from carbon 3 to carbon 2, so this is the 2 phosphoglycerate. This process is called isomerization because the molecule is formula is same. Now next step, which is the ninth step, what happened, the water molecule from 2 phosphoglycerate removes, it's removed, it's go away, it will become phosphoenol pyruvate. Okay, so now we have two molecules of phosphoenol pyruvate. Okay, now this is the 9th step. What is the 10th step? Then now up till now we don't get any advantage from this process, because we give two ATPs, we take the ATPs. Now we are going to take the advantage. Now from phosphoenol pyruvate, two phosphate group is removed, it will become pyruvate, and the phosphate group attached to the ATP, so it will become ATP. So now two molecules of ATP are made. This is the end stage, this is called ATP formation. So this is the process of glycolysis. This is a 10-step process. At the end of the process, we will get from the glucose pyruvate and two molecules of ATP and two molecules of NADH. Okay, so two molecules of water also remove. Okay, so this is the process. Now this all process occurring in the cytoplasm. Now what happened? Need to oxidize this pyruvate. Okay, need to oxidize this pyruvate. The process called oxidation of pyruvate. Now here is another diagram I have to explain you. This is also called link reaction. This is also called, we also call it as a link reaction, if I'm not wrong. This is called link reaction. Why it's called link reaction? Because it connect the process of glycolysis to the Krebs cycle. Okay, this is the link. Okay, what happened in this? The pyruvate goes to the, okay, let's talk like that. What happened in this? This is a pyruvate. The pyruvate, okay, what happened? The pyruvate is basically this is a 3-step process. The first step is decarboxylation. From the pyruvate, carbon dioxide removed from the pyruvate, and it will become acetaldehyde. It will become acetaldehyde. Okay, it will become acetaldehyde. Then this is called decarboxylation. In the second step, from the acetaldehyde, there, this NAD positive are present in the cytoplasm. They are also present in the mitochondria. What happened? This NAD positive takes the hydrogen from the pyroacetaldehyde, and it will become NADH and H positive, and this acetaldehyde become acetyl group. Okay, this acetyl, in the third step, there is a coenzyme A which will attach to this acetyl group to becomes acetyl CoA. Okay, so this is the 3-step process. I write it over here like decarboxylation, pyruvate become acetaldehyde, then oxidation, the acetaldehyde, basically acetaldehyde remove hydrogen ions, and it will become acetyl group, and hydrogen attached to the NAD to become NADH. Okay, so we have two pyruvates, so two NADHs are formed, two acetyl groups are formed, and at the end we will have the coenzyme A attached to the acetyl group become acetyl CoA. This will use in Krebs cycles. I hope you understand well. I will meet you tomorrow with Krebs cycles and electron transport chain. Okay, please do comment you like my video or not. Whatever topic you are weak in, just give me the comment. I will make a video for you. Thank you. Bye.